Hello and welcome, dear professional students and learners, to the interesting topic on stability testing, that is, stability testing for new drug products. This is as per the ICH Q1A guideline, and this video is a part 3 of the stability testing video series. So, as we know, the stability testing is required, or stability studies are required to provide evidence that how the quality of the drug product varies with the time under the influence of variety of environmental factors such as temperature, humidity and light. Then stability testing is also required to establish the shelf life and recommended storage conditions for the drug products or formulations. It is also a mandatory requirement for the registration. Stability data package for the new drug substances or new drug products, that is sufficient for registration application within the three regions of the EC, Japan and United States. So this guideline discuss about the requirements of stability study for the US, Europe and Japan. The choice of the test conditions defined in this guideline is based on the analysis of the effects of climatic conditions in the three regions of EC, Japan and United States. The mean kinetic temperature in any part of the world can be derived from the climatic data, and world can be divided into the 4 climatic zones from zone 1 to 4. This guideline addresses the climatic zone 1 and 2. So the mean kinetic temperature is the temperature which is derived from the Arrhenius equation. This is the Arrhenius equation, and from this the mean kinetic temperature is derived. Based on this, the world is divided into zones. So what are these 4 zones? We will see. So ICH stability zone 1 is in the blue color. Here you can see the ICH zone 1. Then below that there is a ICH zone 2. This is in the green color. Then zone 3. Here zone 3 is given in the orange color, and zone 4 it is given in the red color. So you can understand that after zone 4, here is the India. It comes in the zone 4. So above this it will go to zone 2, and above that the European countries are there. These are in zone 1, but some of the countries are in zone 2 also. So zone 1 is temperate zone. The countries like Germany and European nations and Russia come into the zone 1. The long term condition is 21 degree 45 degree 45% RH. So 21 45 is the long term stability condition for zone 1. Then zone 2 is there, which is Mediterranean zone or subtropical zone. USA, Australia, Egypt, France, Greece and Japan and other countries are also there, which are under the zone 2. So long term condition is 25-60 for zone 2. Then zone 3 is hot and dry zone. Here you can see Iraq, Botswana and Jordan is there. Long term conditions are 30-35. So here temperature is increased while the humidity is decreased. So that's why it is referred as hot and dry zone. You can remember easily from understanding these all the zone conditions. Then zone 4 is divided into zone 4A and B. So A is hot humid, that is tropical zone. The long term conditions are 30 and 65. So long term condition is 30-65. Then coming to zone 4B, this is hot and higher humidity than this one. So 30 degree is there, similar temperature, and the RH is increased from 65 to 75. So if you do the long term stability from 25-60, 30-65 and 30-75, you can cover the entire regions or many of the countries. Then what are the requirements for stability testing for the regulatory filing for the formulation and approval? So number of batches are at least 3 batches, and one batch is required to be tested for photostability testing. So for photostability testing, at least one primary batch is required, and the 3 batches are required for formal stability testing. That is, formal stability testing, 3 batches are required. The scale should be pilot scale, and if you go through the guidelines, you will see that 1 lakh units is the requirement for pilot scale. So separate video is there on exhibit batch scale. If you see that video, you will get the complete clarity. Here we will not discuss that topic in detail. So scale is pilot scale, 2 batches pilot scale, and 1 smaller batch can be there with justification. But generally the applicants make all the 3 batches in the similar batch size, and this is required for all the strengths. Then manufactured by final method and composition. So the composition and manufacturing process should be final, which is the representative of or proposed of same commercial manufacturing batch of the formulation. In these formulation batches, that means 3 formulation batches, different lots of APIs are required to be used. This is the requirement to understand the effect of API lots on the stability and variability of the process. So some of the applicants are using minimum 2 lots, some are using 3 lots. Then if the container closure system have the impact on product performance, then different lots of packaging material or devices are also required to be used. Then strengths and pack sizes. So for example, you are making a formulation of 3 strength, 300 mg, 200 mg and 100 mg. So so all strengths and pack sizes are required to be manufactured, and all strengths and pack presentations are required to be loaded on the stability. Then all the strength should have the scale of pilot, and number of batches are 3, and each strength should undergo photostability testing. That is must. Bracketing and matrixing can be applied. So bracketing and matrixing designs and principles are used to reduce the stability samples mainly. So bracketing and matrixing can be applied, and justification is required to be provided. Then coming to the container closure, as we have seen, the container closure should be same or simulates the packaging, primary and secondary packaging along with the label. These are the minimum requirements which are proposed for storage and distribution. So the container closure should be same or simulates the packaging including primary and secondary and label, which is proposed for storage and distribution. Specifications. Then specification should include all the testings parameters which are affecting the safety and efficacy of the formulation. So it includes physical, chemical, biological and microbiological attributes, including the antimicrobial efficacy and functionality testing, which are specific to the drug product to be studied, as these affect quality, safety and efficacy. For example, the functionality testing like uh, activation for the metered dose inhalers or dose per activation, these are the functionality testing, and these are required to be included in the specification. The analytical method should be stability indicating. Release and shelf life specifications are required to be in line to the ICH guidelines, and if different specifications are given to the release and shelf life, then its justification is required. Then coming to the testing frequency. So all these 3 batches are required to be loaded on long term, accelerated and intermediate storage condition. So for long term, establish the shelf life. This long term condition is required to establish the shelf life, and at least 12 months is the requirement. So coming to the frequency, every 3 months over the first year, that is 0, 3, 6, 9, 12 months, every 6 months over second year, and annually thereafter throughout the proposed shelf life. So if the proposed shelf life is 3 years, then from 3 years starting, it will be tested annually. Then accelerated storage condition. This is the requirement for extrapolating the shelf life. At least 6 month data is required. 1, 0, 3 and 6 months. If there is a significant change expected, then we have to include the fourth time point. Like we have to increase the sampling in this uh, accelerated storage condition, like 1, 2, 3 and 6 months, as sometimes the research phase, that is during the research we found that this product may generate impurity, for example at accelerated state. That time the fourth time point can be included. Then coming to the intermediate storage condition, if significant change at accelerated storage condition is there, then the intermediate storage condition is tested. And while loading the batch on stability, this intermediate storage condition is loaded. Minimum 12-month data is required for this intermediate condition, and minimum 4 time points are required, including the initial and final time points. For example, 6, 9, 12 months. So including initial, it will become 4 time points. At the submission time, 6-month data from 12-month data is required. Here also, reduced design like matrixing and bracketing can be applied to the product. Separate video is available for in-use stability testing. You can go through that video and have the complete understanding about what is the in-use stability testing requirements. Then long-term condition, 12-month and continued for a period of sufficient to cover the proposed retest period. So it is not only the 12 months period for stability testing, it is the period for long-term stability testing till which the applicant is going to propose the retest period or shelf life period. Data from the accelerated storage condition and if appropriate from the intermediate storage condition can be used to evaluate the effect of short-term excursions outside the label storage conditions which might occur during shipping. Then storage conditions. So long-term condition, intermediate condition and accelerated condition is there. See, always 4075 will be the accelerated condition and it will require 6 months. Then intermediate will require 6 months, but the applicant are not forced to 6 months only. We can load this intermediate condition sample till 12 months so that we can have the complete understanding of the product. Then long term is 12 months requirement until the completion of the shelf life period or expiration period, or that is also known as retest period. So the condition is 25-60 degree, 25 degree and 60% RH, or 30-65. If long term condition is 30-65, then no need to test the product for intermediate because both these are the same. So it is up to the applicant to decide whether the long term stability studies are performed at 25 and 60 or 30-65. And if 30-65 is the long term condition, then there is no intermediate condition. Then coming to the significant change. Now you have loaded the stability batches and you got the data. So when you will say that there is a significant change or the batches are passing. So significant change is the very important term in stability testing. And significant change is nothing but any deviation or any failure for the batch in any of the specified parameter or specified test. Even if the product fails in appearance, that is also a significant change. If you load the white tablets into the stability and at the end of 6 months accelerated storage condition, the tablet becomes slightly yellowish in appearance, that is also significant change because appearance is changed. Here I have given some examples from the ICH Q1A that change in 5%, a 5% change in assay is a significant change. Any degradation product exceeding the acceptance criteria is a significant change. So changes to the pH, failure to meet the acceptance criteria of dissolution for 12 units, that is also significant change. So these are some of the examples. Changes which are not considered significant. That means like softening of the suppositories at elevated temperature of 40-degree accelerated condition. That will not be a significant change because that is very much normal. The composition of suppositories is such that it will become soft. Melting of the creams at accelerated condition and cross-linking of the gelatin capsules. These are not considered as significant change. Then stability testing. So some of the products are packaged in the impermeable containers. Some of the products are packaged into semi-permeable containers. So the products which are packaged into the semi-permeable containers, for those the stability conditions are given. And for semi-permeable containers product, we have to test the product at low humidity conditions. So long term is 25-40. Here the humidity is lower, and 30-35. Then intermediate is 30-65. Accelerated is 40 and not more than 25% RH. So this is accelerated. This is long term. But here you can see the intermediate condition is 30-65. The exact reason of this I have not understood it, but still you can consider that if the product is stable at 25-40 and 40-25, then this is somewhat different type of intermediate condition. Then drug products are packaged in semi-permeable containers. A significant change in water loss at the accelerated condition cannot be considered as significant, and it does not necessitate testing at the intermediate storage condition. However, data should be provided to demonstrate that the drug product will not have significant water loss throughout the proposed shelf life if stored at 25 degrees Celsius and a reference relative humidity of 40% RH. 5% loss from the initial value is considered significant if the product is packaged in semi-permeable container after the equivalent of 3 months storage at 40-25. So these are some of the examples of semi-permeable containers, and for these type of containers the lower humidity conditions are required for stability testing. Then products intended to be stored in refrigerator, that is 2 to 8 degrees Celsius. So long term will be 2 to 8 degree and accelerated will be 25-60. And drug products which are intended to store below -20, these product will be treated on case by case basis and depending on the product. Then what are the stability commitments? See, the applicants will load the batches on stability, but the applicant may or may not have the complete data of 12 months or as per requirements, but 6 month data he should that applicant should have at the time of submission. So when available, long term stability data on primary batches do not cover the proposed shelf life granted at the time of approval, a commitment should be made to continue the stability studies post approval in order to firmly establish the shelf life. That means though the applicant files the product, still the commitment is required to continue the stability study post approval in order to firmly establish the shelf life. So this commitment is required that the applicant will do the shelf life stability studies, where the submission include long-term stability data from 3 production batches covering the proposed shelf life. A post-approval commitment is unnecessary. So if complete data is there, then no commitment is required. Otherwise, one of the following commitments should be made, and these 3 commitments are there. These are based on the availability of data. If the submission includes data from stability studies on at least 3 production batches, commitment should be made to continue the long-term studies throughout the proposed shelf life and accelerated studies for 6 months. Then if the submission includes data from stability studies on fewer than 3 production batches, that means batches are less than 3 batches, Commitment should be made to continue long-term stability studies throughout the shelf life and accelerated studies for 6 months, and to place additional production batches to total of at least 3 on long-term stability studies throughout the proposed shelf life and on the accelerated studies for 6 months. This is nothing but the commitment given by the applicant to regulatory authority that the applicant will continue the stability studies. Then third, if the submission does not include stability data on production batches, a commitment should be made to place the first 3 production batches on long-term stability studies through the proposed shelf life and accelerated studies for 6 months. The principles of formal stability studies are applicable to commitment batches also. So whatever the principles are enlisted by ICH Q1A for drug formulations or drug products, these are also applicable to the commitment batches. Then stability evaluation. Now you have loaded the stability batches for the formulation and you have a data. Now how to evaluate that stability data? So that we will see now. A systemic approach should be adopted in the presentation and evaluation of the stability information, which should include, as appropriate, results from the physical, chemical, biological and microbiological tests, including particular attributes of the dosage forms. So the meaning of this is you should evaluate and present all the data and the attributes of dosage forms like dissolution data for the oral formulations. The purpose of the stability study is to establish, based on the testing of minimum 3 batches of the drug product, a shelf life and storage and label storage instructions applicable to all future batches of the drug product manufactured and packaged under similar circumstances. This means that based on the data of 3 batches, the future batches fate is determined. The degree of variability of individual batches affects the confidence that the future production batch will remain within the specification throughout its shelf life. So at the time of application, the applicant will have the data of 3 batches only, and that 3 batches data should give the confidence that the upcoming future batches will comply to the specification. Where the data show little or very little variability or little degradation, that is apparent from looking at the data that requested shelf life will be granted, it is normally unnecessary to go to the formal statistical analysis. Providing a justification for the omission should be sufficient. So if the data of the formulation throughout the stability is with less variability and very normal or little degradation is there as expected, then there is no necessary to go for the formal statistical analysis. An approach for analyzing data of quantitative attribute that is expected to change with the time is to determine the time at which the 95 one-sided confidence limit of the mean curve intersect the acceptance criteria. So the simple meaning of this is, if you have variability in your data and if you are extrapolating the shelf life from the available stability data, that time the statistics is required to be used to determine that what will be the time limit or shelf life period at which the product will meet the acceptance criteria or it will fail in the acceptance criteria. That acceptance criteria may be for impurity, maybe for dissolution or assay, anything. Usually the relationship can be represented by linear, quadratic or cubic function on arithmetic on logarithmic scale. So these are all the statistical part, and statistics is used for the extrapolation of shelf life. Statistical methods should be employed to test the goodness of fit on all batches and combined batches wherever appropriate to the assumed degradation line or curve. So all the statistical part is given in the ICH Q1D guideline. So separate video will be there on stability evaluation. Then limited extrapolation of the real-time data from the long-term storage condition beyond the observed range to extend the shelf life can be undertaken as appropriate and justified. So justification is required that should include the complete data, the information about the batch, why this why the degradation is there or decrease in the assay is there, what are the mechanisms, these all points should be discussed. Then any evaluation should not consider only the assay but also the degradation product and other appropriate attributes. So while extrapolating and evaluating the stability data, all the parameters in accordance with the relevant national regional requirements. So storage and labeling is given on the basis of the stability data, and the terms such as ambient condition and room condition should be avoided because this doesn't give the true values for storage temperature and condition. There should be a direct link between the level storage statement and the demonstrated stability of the product. So based on the stability data, the storage can be given and expiration dates should be displayed on the container label. So I hope you might have got a good understanding about the stability testing for new formulations or new drug products. The videos are little bit lengthy, but taking into consideration the guidelines and the length of the topic, this is very much important to cover each and every point. So I hope you might have got some more information and understanding out of these videos. See, guidelines are always there, but it is very difficult to understand the guideline in limited time. So these type of videos will help you to understand the guideline in detail. Many videos are there, and go through those videos from the Pharma Learning in Depth channel. Please do like, share and subscribe. Thank you.